So now we're going to start going through each one of these processes and in more detail and saying what actually happens in each part. So we're going to start first with glycolysis, and then I'll recap in the next video, and we'll go to the next step, etc. So that's how we're going to go. All right, so let's just talk about glycolysis. Now you can see this is a 10-step process. You do not have to know the names of all the molecules in all 10 steps. What I want you to do, though, we're going to go through the process, and I want you to see generally what's going on, and then what we started with, what we ended up with, and what was used in the process. Okay, so let's just go through it one thing at a time. Okay, so first of all, they always use glucose as the precursor molecule. Now the reason why they talk about glucose is because glucose is a molecule that goes through all the steps of glycolysis. In other words, if I had started with another molecule, let's say for example fructose, I might not have gone through the entire steps of this process. So that's one reason, and the other reason is that some disaccharides or polysaccharides have to be broken down first through hydrolysis in order to get the glucose molecules or the sugar molecules that can then enter the process. So once we are in glycolysis, we have to start with a sugar, and the sugars that go through the entire process are glucose. All right, so that's why they use it to talk about. Now notice this, let's look at this molecule really closely. Now, if you might not recall, but if you, just to kind of point out, that it has 6 carbons in the ring. The carbons are not represented in this way of drawing the molecule. In other words, these are the corners of the actual ring, and each corner is a carbon, and it starts numbering over here. So that's 1, 2, 3, 4, 5, and the 6th one is hanging off. So I kind of think of this like a little handle that is hanging off the molecule up there. All right, so the very first thing that happens in the process is that ATP adds a phosphate group to the handle. So you see that? It added that phosphate group there. And then the second thing that happens is that the molecule gets rearranged. Notice that in order to break this molecule, basically what's going to happen in this process is I'm going to end up breaking this glucose molecule in half. So I'm going to go from a 6-carbon piece to 2 3-carbon pieces. So that's ultimately where I'm going to go here. If I have 5 in the ring and a 6th one hanging off, what I have to do is then make that molecule symmetrical. In other words, I have to have 4 in the ring and 2 hanging off, right? So that's what happens in this step. And notice that there's enzymes for each step, and they end with the term ACE, and that's how you know, oftentimes anyway, that that's an enzyme. Okay, so the next thing that happens is, you might have guessed, is I'm now going to phosphorylate the other handle. So now I have a more symmetrical molecule, and this is the last step before I can literally divide it into 2 equal pieces. At first they're not completely equal, but the enzyme isomerase can make 2 of these called glyceraldehyde 3-phosphate. That is, the phosphate is added to the 3rd carbon group. Okay, so anyway, this I'm going to end up with 2 of those. So the whole first part of the process is I'm adding ATP in to phosphorylate my handles, that I can then, and making the molecule symmetrical so I can divide it into 2. Okay, so the next thing that happens, and this is a little bit confusing I think in the textbook, because this whole diagram is ×2. And what they are showing you here, so they're saying 2 of those, but the actual amounts that are here are for, there's 1 for each. So these are for both of them, and this is just showing you 1 of them. So the things in white are the 1 parts, and the things in, outside of that are showing you the actual numbers for 2. Okay, so anyway, I have two glyceraldehyde 3-phosphates, or G3Ps is what they're sometimes called. And I'm going to enter those into the next part of the process. So the first thing that happens is I'm going to take off some excited electrons from both of those glyceraldehyde 3-phosphates, or the G3Ps, okay? So I'm going to take some off. I'm going to get then some NADH. NADH is carrying excited electrons. Also, during that process, I add some phosphorus in. This is basically a type of phosphorylation, holding the energy really in that molecule, all right, or using the energy from that molecule to do that, okay? Now what happens is I'm going to take off the phosphate group, or take off one at least, and I'm going to take off one per molecule, that's why I end up with two, and make ATP out of it. And then I'm going to change configuration just a little bit again, a couple of times, and I'm going to then take off another ATP from each one, or two, so I end up with 4 ATP here. Originally I put in 2, and I get 4 ATP from the process. I also get 2 NADH, or 2 molecules that are carrying excited electrons. And the outcome of the process is pyruvate.